Hello guys, there is the another video in which we interpret the climate indication through the biomarkers. As we discussed early in two videos, we can be interpret the climate through carbon isotope and through fossil record or through any other proxies like rings like oxygen isotopes and carbon isotopes. Now in this video we can explore the biomarker, the biological response to the climate variability. It is a very important topic, and it also important topic for the climatic change. They are the very recent proxy to interpret the climate change by biomarker, and what is biomarker and how they are can be changed through climate variability. So we now discuss in this lecture. So first of all, what is biomarker? Biomarker is the organic compound in geological and biological material that indicate the past environmental condition. It is also known as a molecular marker or geochemical fossil. Basically, these biomarkers are present in the rock record from the Precambrian to the recent rocks, and by analysis these rocks in between the biological response, we can interpret the paleoclimatic change. So there is the two types of biomarker: molecular fossil and lipid biomarker. Molecular fossil include like styrene, hopen, which are mostly linkage to the bacteria, and while the lipid biomarker such as alkenones, long chain aliphatic hydrocarbon, they are mostly associated to the algae and plants and terrestrial output or terrestrial environment. These both molecular fossil and lipid biomarker are specifically related to fats, and both help geologists to understand the ancient life and environment. And by analysis the ancient life and environment, we can easily interpret the climatic change, how they are the climatic, how they are the ocean, where is the temperature, how they are precipitation, how they are the sun rays, is how if there is the oxygen level is high, if the carbon dioxide level high, low. So by analysis the molecular fossil or lipid biomarker, we can be easily interpret the past climatic change. Their implication is various. For example, by identify the organic source rock which are present in the shell of the rocks, they can be interpreted the million of year ago ecosystem like temperature, salinity, and oxygen and depth. Biomarker are particularly also useful for the source of organic matter. Where is this organic matter is come from? If they are the heterogeneous organic matter, means they are allochthonous, they are transported, so they can be show you the source of the organic matter. The petroleum exploration uses the biomarker like hopen, styrene. They trace the origin of the crude oil, where is the source rock and where is they come from. By analysis the source, they can be interpret this that the reservoir will be high or low. The geochemistry and paleoclimatology, organic matter, they reconstruct the past climate or ecosystem by analysis the biomarker like alkane, isoprenoid. They understand the past biological environmental condition. So we will be later on discuss how they are interpreted into the past climate. So there is the figure which can be shows that within depth the organic matter, they can be generate the oil by from the source rock at 3 to 4 kilometer. It is the mature zone, and whenever they are move through migration, the petroleum, they they move to the reservoir rock. So by analysis the reservoir rock hydrocarbon, we can interpret this that what is the source of the petroleum reservoir rocks, and the proxy is obviously biomarker. So the biomarker environmental studies started from the 1990s, and various works are present, like in physiological biomarker, like it include immune response, growth and reproduction of the tiny organism, and they can be interpreted by to impacted by the climatic change. The other is genetic biomarker, investigate the genetic material to identify various gene that indicate adaptation or response to the environmental changes. So this is the graph which can be shown that from the 1880s to 1990s or 2000 or 21st century, they all work are done on the publication of the climatic change. But after the 2000, the proxy, like the biomarker, has started, and various publication is published, and up to now 160 publication is published on biomarker and the climatic change. But to 1880 to 2020, almost 30,000 publication is published on the climatic change by not involving the biomarker proxy. So this is the new method, and various research is needed for the biomarker analysis, and they are related with the climatic change. So this is the rank which country is mostly spend their money and spend their knowledge in the research of the biomarker. So the first number is United States, and second is China, Germany, and the ninth is Italy. And this is the various journal or various academies where the biomarker analysis is mostly focuses and mostly research on it. Like first is the Chinese Academy of Science, and this is the list, and this is the country, and this is the number of publication. So this is the overall world map where is the biomarker study and how much publication is done. So first number is America and China. This circle is showing the publication rate. So there is a high publication rate, and this color shows the which country is more research on the biomarker analysis, and they related with the climatic change. So this is the joint collaboration of different countries. The red one shows that these are the mostly most publication work on done by the climatic change biomarker analysis by this country, and this green one is the second number, and the third one is the blue color. So this is the various sources in which the biomarker is present, and biomarker is research on it. So for the geologist, the organic geochemistry is the main source in which they research on the biomarker analysis and the relation with the paleoclimatic changes. This is the two cluster, one is the red and one is the green, in which different research is done on the basis of different field. Like for the geology, paleoceanography, organic geochemistry, they are all related to nature, and they correlate research work on the basis of the biomarker through this field. And this is the green field is for the agriculture purpose, like soil strength and genetic analysis of the crops, and they are related with the science. So these two clusters just showing the different field in which the biomarker analysis can be interpreted. This is the various biomarker and various different tiny organism of different microfossil which can be remained there biomarker like this structure. So this is the diatom biomarker, highly branch isoprenoid. Whenever this structure can be found in the rock, so it means that this is a diatom biomarker. This is the dinoflagellate. Dinoflagellate are mostly shows the marine environment. This is the crude oil. This is the I think this is the angiosperm. plant biomarker, and this is the ratio of the Hoppen's terrain. uh, if you go to the higher ratio, Hoppen to terrain ratio, it will be give the environment of the lacustrine or terraneous environment. If you back to the low ratio, it will be give the marine or saline environment. So this is the age diagnostic biomarker like Hoppen, styrene, and isoprene, pristine, and phytine. So whenever there is the high in the styrene, so it gives the Paleozoic to Cenozoic age diagnosed. Whenever you can see the uh, terpene, so it will be give the after the Cretaceous strata. If you have the biomarker of the isoprene, so it will be give the Jurassic to the Neogene age diagnostic biomarker. So this Hoppen is the basically this bacteria cell membrane, and they have present in the sedimentary rock, and this is the structure of the biomarker of the Hoppen. This is this structure of this terrain, and this is the isoprenate structure of the two pristine and phytine, which is the part of the chlorophyll. When they are the diagenetic process occur of the chlorophyll, so they are converted into a pristine and phytine. So whenever you can see the pristine and phytine, so it mostly associated to the chlorophyll environment, in which you can see there is a high temperature, a high productivity. If you see the terrain biomarker, so it will be give the terrestrial plant, terrestrial organic matter, the environment interpreted by the biomarker. So this is the process how the biomarker can be studied. So first of all, there is the tiny organism present in the ocean. When they are die, the sediment layer are covered, and whenever we just see it, so they have the cell like this one. So they have DNA, cell wall, photosynthetic lamina, and the chinosome. So this chinosome can be chlorophyll. If they this is the biomarker. If this chlorophyll is the linkage of the of this tiny fossil, and this one molecule can be naturally degraded into the two different compound like pristine and phytine, which can be discussed early of the isoprene biomarker. So whenever you see the pristine phytine, it means that this is the remnant of the life. So if you found in the Mars, so it means that this pristine and phytine is the remnant of the life, and there is the life existent in the Mars. So this we can be focused from the Cretaceous age of the Pakistan for the biomarker analysis. First of all, if the styrene and Hoppen characteristic ratio will be high, so it means that it will be the marine organic matter. If the styrene and Hoppen ratio is low, so it will be give the heterogeneous environment. If you back to the previous slide, so it is the Hoppen slash styrene ratio. So if they are high, so it will be give the heterogeneous environment. It is the Hoppen slash styrene ratio. If you go to the this slide, so it is the styrene slash Hoppen. If they are high, to it means it will be marine organic matter. So ultimately, if Hoppen slash styrene ratio is low, so it will be give the marine environment. If the Hoppen slash styrene ratio will be high, so it will be mean that it will be give heterogeneous environment. Or if you just say that the styrene slash Hoppen ratio is low, so it will be give the heterogeneous environment, which means that the Hoppen concentration will be the styrene concentration will be low, so it will be give the heterogeneous environment. The they where they form the Cretaceous rock. So in Pakistan, the Cretaceous rock are distinguished into two basins, the upper Indus basin and the lower Indus basin. If you go to the upper Indus Basin, it have Chichalilam Shival Kawagar formation of the Surghar ranges of the Surghar group of the Cretaceous strata. If you go to the lower Indus Basin, so it have symbol Guru Par, Bogal Coat and Forbondro formation. So we mostly focus the lower Guru and Simber formation of the Cretaceous strata. So they have high maturity level. Why they are high maturity? Because of the analysis of the biomarker. They have contained highly sterane, it means abundant heterogeneous organic matter, while the par and guru formation are immature, so they mostly cover into the all window zone because the sterane ratio is low and they are mostly marine environment. uh, Other biomarker like oleanins are also present, it gives the hint of the age. You can see the age diagnostic that oleanin are present in the Cretaceous strata, so it means that the Guru and Sember formation are at the Cretaceous strata. They can also be checked by the study of the uh, planktonic foraminifera. If you just see the planktonic foraminifera of the Guru and Sember, it will be interpret the Cretaceous strata. What is the olein? Olein is the derived from the angiosperm land plant, and they are dominated by the land plant which are present in the late Cretaceous. So this is the biomarker of molecular fossil structure of the oleanins, and this is the peak of the 50 to 52 million year age of timeline. Which can be clearly shows that the in the Cretaceous strata the oleanin concentration is very high. This is the outcrop of the Cretaceous strata of Pakistan Guru formation. It is the anoxic environment which started from date 1. This is the Cretaceous anoxic event of 1, which is known as Boronelli event. In the Cretaceous strata, there is the pyrite nodule which shows the anoxic environment and also show the oxygen deficient environment. This is the OA2, another oxygen anoxic event which are present at the above part of the Guru formation. This is the Simba formation, which is the darker color shell, and they are the heterogeneous environment. Means the Hopen sterane to Hopen ratio will be low. If there is low sterane to Hopen ratio, which means that it is a heterogeneous environment, and this is the par formation in which the sterane slash Hopen ratio will be high, and this is the Guru formation of the upper part of the lower part. So future research trend in the biomarker. Biomarker is mostly focused in the future in the past climate because it's the best proxy for the past climate. They are also studied the soil strains by which improve the crop, and they are also studied the marine environment in which mostly focus to like fish organism to explore the biomarker operation growth and health, looking for the treatment which are mostly affected due to the climatic change. Biomarker in polar and high altitude ecosystem have also gained increased importance due to the distinct challenges posed by the climatic change in these organisms in this region. Uh, what is a paleoclimate? As we discussed that the biomarker is mostly associated with the past climate. So what is the paleoclimate? The study of the past climate using the proxy like tree rings, ice cores and biomarker is mostly associated with the paleoclimate. This is the various proxies. By various proxies we can be helped to understand the natural climate variability, and also give the modern climate changes by study the past climate. It also understand the ecosystem and species response due to the climate change, and also reconstruct the past ocean and atmospheric circulation. So in this figure we just see this is the chemical proxies like oxygen isotope, silicon isotopes, nitrogen isotope, biomarker analysis on or carbon isotope. Physical proxies like tree rings and various outcrop lithology like black shale can also interpret the past climate. Biological response like we studied the foraminifera and then section analysis and rock analysis. And this is the various proxies like ice core, tree rings, corals, and evolution and sedimentary core and stalactite and stalagmite of the cave. So they are all the proxies in which we study these proxies, interpret the past climate and modern climate. What will be the changes? So this is the whole world map in which various field the corals, lakes, cave, macrofossil, tree rings, and ice core will be studied. And by studied date, we can interpret the past climate like that one, that what is the climate from the 1,000 year before, and what will be the climate of the 100 year after the 2024. So why biomarker mostly uses for the climatic change? This is the six reason. Number one is chemical stability. Uh, Temperature, like temperature, pH, microbial activities, they are mostly resistant. They are not changed from the temperature, pH, and microbial activity. For example, like lipid such as alkenones, sterol are common stable biomarker that persist or resistance in sedimentary over long period. Uh, Another is the consistency in biological origin. They are remnant consistent throughout the history, like algae, bacteria, or plant, means they can be not involved or not changes due to the environmental effect. They will be consistent and give the accurate knowledge of the past climate. The other is the preservation of the natural record. Preserved in ice core, sediment, tree rings, and fossil remains. They are mostly and all of the proxies, the biomarker will be present. Minimal alteration over time. They will be diagenesis due to burial and every like temperature and pressure and pH. So there will be minimum alteration, means give the actual environmental changes, shows the past climate. Another is when there is a sudden change in the environment, they will be give the exact changes why the sea surface temperature is high or low. So scientists to reconstruct past climate easily. And applicable in different system, means they are not restricted to the marine. They can be showed to the terrestrial rock or atmospheric, making reliable indicator for tracking changes in the ecosystem and environment across the million of years. If you found the marine environment rock like limestone, dolostone, or uh, like deep marine shale, so you have the biomarker and you can analyze the paleoclimate. If you have the fluvial system like sandstone, conglomerate, siltstone, or clay, so it will be also give the climatic change. If you have the atmospheric oxygen isotope, carbon isotope, so you can interpret the biomarker easily. So this is the types of biomarker: terrestrial biomarker, marine biomarker, isotopic biomarker. Terrestrial biomarker like mostly lignin, mostly derived from the terrestrial plant organism like leaf waxes or the branch GDGT, mostly from the soil bacteria and pollen spores. So if you found these biomarker, it will be interpreted directly that they are from the terrestrial biomarker. Marine biomarker like mostly preserved in the algae, bacteria, and marine plant. The terrestrial biomarker mostly present in lake beds and floodplain of the fluvial system, while the marine biomarker mostly preserved in the algae biomarker, bacteria, and marine plants. Like alkenone, they are hypo to fight marine algae mostly present in the in the ocean, and they give the sea surface temperature reconstruction. Sterol or isoprenite GDGT, this is the branch GDGT from the soil bacteria, and this is the isoprenite, which is the chlorophyll bacteria, and they are also water sensitive, give the paleoclimate water temperature. And pigment like chlorophyll also give the marine biomarker indicator. Isotopic biomarker mostly present in the carbon and hydrogen isotope, and they are present in the sediment, ice core, and tree rings. For example, if you find the plant and you can be focus on the isotopic analysis, they will be give the environmental condition, like whether the climate was dry or wet. This is the biomarker of hydrological changes. Also give leaf waxes, which are mostly heterogeneous environment, and they give the either there is the monsoon, or there is the hereditary, or there is the ancient rainfall. So they will be reconstruct the past precipitation, humidity. For example, isotope like deuterium, which is the hydrogen isotope, if you find it, for example, in a high value of the deuterium value of the hydrogen, so it means that there will be arid region, because hydrogen 1 is evaporated and hydrogen 2 will be left. So it means that the evaporation process is very high, so it means the temperature is high and the aridity will be automatically high. So this is the very best diagram in which you can show the different biomarker which show the different environment repetition and give the hint of the different environmental climatic changes. So by this all the biomarker analysis, we can reconstruct the past climate and plus past environmental reconstruction of the climate. So this is the biomarker for the carbon cycle by study the sterol and terpenoid and isotopic signature. By this study, we can be interpreted the what is the past climate. For example, you can see the Pangaea and the overall ocean of the Pentalasa. If you study the biomarker of the carbon, so it will be give the cold climate. For example, biomarker of isotopic analysis, for example, the shift of the C3 plant to C4 plant in Cretaceous strata, it will be studied from the biomarker. For example, increased C4 plant means it will be higher the carbon 13 ratio. In the sedimentary core may indicate it will be grassland. C4 plant are the mostly tree, shrub, and highly forest environment, which means that it will be high rainfall, high water concentration. If you switch to the C4 plant are mostly the grassland plant in which no water needed and very dry environment. So from Precambrian to Cretaceous strata, all of the C3 plants in which the carbon 13 value is low because the carbon 12 ratio will be high. So by this reason, the carbon 12 ratio is high. So that mean that C3 plant will be low the carbon 13 isotope analysis. And in C4 plant, the carbon 13 ratio will be high because the C12 carbon 12 isotope will be low. So by analysis this complex isotopic analysis and biomarker, we can interpret the past environmental reconstruction. So this is the, for example, two stages in which the PETM Paleozoic to Eocene, which are mostly present in Pakistan, like Patala Formation, 56 million years ago. In this environment, the climate are rapidly warm, and the last glaciation they are rapidly cool. So by study the biomarker like organism of the bacteria, this is the name of the bacteria. If they are present in the sediments, it means that the temperature is really very high, because these are the bacteria in which they are highly resistant bacteria. So they are so they can be give the clue of the very high temperature. Alkenones are mainly and marine algae calculated for also suggest the SST sea surface temperature, in which for example if they are the alkenone very long chain, so give the increase of the SST. If they are the decrease of the long chain, means the double bond are mostly present, so so it will be give the low temperature. So in the PETM, so the double bond will be fewer, but the long chain will be high because of the high temperature. uh, Life plant leaf like as we discuss C3 C4 plant. The C4 plant to C4 dominant will be uh, increases because the aridity will increase because of the temperature increase. If the temperature is low, the C3 will be dominant. C3 means forest will be dominantly mean highly precipitation environment. If the temperature is really high, so it means the evaporation will be high, so it means the C4 plant will be high. So it will be in contrast, alkenone double bond will be mostly present in the temperature cool environment. The biomarker of the leaf decreases precipitation, so it means the shift to the C4 to the C3 plant. Change from the C3 to C4 plant indicated the dry condition and less precipitation. So whenever there is the less precipitation, it means the C4 plant will be increases. The evidence on the low marine environment will also like sterol concentration will be decreases. This is the biomarker which indicate the temperature cool environment and low precipitation. So this is the limitation. Also limitation occur in the biomarker like diagenesis. If there are many microbial activity chemical reaction occur, so the biomarker will be give the less hint to the past climate. Biomarker, for example, regionally specific, limited to the global interpretation. If you work in the Pakistan, so you can also interpret in the Pakistan paleoclimatic analysis. You cannot interpret the whole world climatic change. So this is the limitation, that it will give the local scale information but valid information. uh, Complexity in signal means the biomarker often reflect multiple environment like temperature, salinity, precipitation, vegetation. So you will be complex, that what will be tell either temperature tell, salinity tell. So one proxy or one uh, environment cannot interpret it. It will be interpret multiple environments, so it will be little bit confusing. Because for example, if we grow the plant, so there will be multiple effect or takes place like carbon dioxide, temperature, humidity, sunrise, where is the wind, where is the transportation, where is the respiration, photosynthesis, chlorophyll. So the chlorophyll will be also give the all the environmental, which will be multiple complex thing. So this is the future direction of the biomarker. Is the suggestion that new biomarker proxy will be developed to interpret more good paleoclimatic reconstruction. Also advance in the isotopic analysis, for example, not to very strict to the carbon, sulfur or oxygen isotope. So you will be more and more isotope analysis. Other is integrate will be other proxy. If you study the biomarkers, so you will be just focuses at the other like pollens, reconstruction, trading. Also correlate the other proxies to 100% sure that this biomarker result will be so percent accurate. So thank you. We will be meet in the next video. Thanks for watching.